Right. Hello, everyone. Once again, we are adding the presenters to the panel so that they can answer take the questions. There's one presenter yet to join us, which is NURULUMI AHMAD. NURULUMI, if you're there, please raise your hand so we can add you in. We will start exactly at 11:30. It's already 11:30, so I guess we shall start. All right. Welcome to our poster presentation session. We will have 12 presenters presenting their posters and research. Each presenters will be judged by myself, the moderator, Dr. Izuddin from University of Kuala Lumpur, and another judge, Dr. Shafiq Asnawi from Monash University, Malaysia. The presenters are given 2 minutes as a pre-recorded video to present the poster, and you will be given 3 minutes to answer the questions from the judges. You will each be given one question from either of us, and then you will allow us a few minutes to record your scores. All right, without further ado, I would like to invite the first presenter, Muhammad Asyaari Zakaria from Universiti Kebangsaan Malaysia, to present his research. Please welcome Asyaari.
Assalamualaikum and hi. I am Muhammad Asyaari Zakaria and I'm going to present my poster entitled Role-Associated Kindness Signaling Pathway Activated in Response to Increased Tissue Rigidity in Lung Squamous Cell Carcinoma in Vivo. Recently, increased tissue rigidity was acknowledged as an emerging hallmark of cancer, as it can promote cancer growth by activating ROCK signaling pathway in numbers of cancer. However, their relationship in lung SCC remains elusive. Thus, we aim to determine the rigidity of lung SCC tissue by analyzing epithelial thickness, proliferation status, and collagen content. Next, we aim to determine ROCK signaling pathway activation by analyzing ROABC, ROCK1, and ROCK2 protein expression. For the methods, female Balqis mice were allotted into two groups, which are vehicle and cancer, that receive 70% acetone and anti-CU and SCC carcinogen, respectively, for 30 weeks. After that, we harvest the lung for HNA staining to confirm lung SCC formation and measure epithelial thickness. Then, we did IHC Ki67 and serous rat staining to determine cell proliferation and collagen content, respectively. Also, we did Western blot to analyze ROCK protein expression. Briefly, we found a significantly higher epithelial thickness, proliferation status, and collagen content in the cancer group as compared to the vehicle, which indicate increased tissue rigidity. We also found significantly higher ROABC, ROCK1, and ROCK2 protein expression in cancer group as compared to the vehicle, which indicate ROCK signaling pathway activation. In conclusion, our findings suggested that increased tissue rigidity might be responsible in activating ROCK signaling pathway in lung SCC in vivo. That's all. Thank you for listening.
Thank you very much, Asyaari. I think I will go first. First and foremost, well done on the presentation. The poster looks nice. Just wondering what is the reason for you? Because I'm pretty sure in cancer, um, um, mechanism and pathways, it involves very complex pathways and mechanisms, and it involves a lot of factors and enzymes and whatnot. Why specifically that you choose this particular ROCK signaling pathway?
Okay, thank you for the question. Can you hear me?
Okay. Yes.
We chose the ROCK signaling pathway because it was the most studied mechanism that was activated in response to increased tissue rigidity, because it plays an essential role in counterbalance the extracellular force generated by the increased tissue rigidity. So that is the reason why we choose ROCK signaling pathway.
Okay, in terms of targeting this pathway for treatment, do you see any future intervention targeting this specific pathway?
Yeah, based on the, we haven't done the intervention on this specific pathway yet because we're only looking at the expression and the association. But as I can see from the previous study, there was a lot of studies that showed a very positive impact of inhibiting or suppressing this rock signaling pathway in cancer.
So specifically, which part of the pathway?
Which part of the this rock signaling pathway?
Yeah, specifically which part, because the pathway itself, it has several pathway, right? I mean, like it's not like just a direct mechanism.
I see. So it's for the rock, we can the previous study can be divided into kinase targeting rock and non-kinase targeting, which is target others pathway that was associated with rock. So for the targeting the kinase, which is directly targeting the rock, they show it show a very promising therapeutic opportunities for this in treating cancer, similar to the non-kinase targeting rock.
Right.
So it involves a lot of other pathways
yes.
actually that associated with rock.
Yes, of course. All right, thank you, Asyaari, and well done. Right.
Thank you.
Thank you. Give us 1 minute to record your score. Okay, we move on to our next presenter, Dr. Liza Noordin from Universiti Sains Malaysia. The floor is yours, Dr. Liza.
Hello, good day everyone. I'm Liza Noordin from USM Health Campus, Kelantan. I'm presenting my research project entitled REM sleep deprivation-induced endothelial dysfunction. Oxidative stress is a known cause of endothelial dysfunction. REM sleep deprivation is associated with oxidative stress. So, this study focused on oxidative stress as a potential factor for endothelial dysfunction, also evaluated the protective effects of vitamin C in REM sleep deprivation. We used male spread dolly rats that were divided into 4 study groups as shown in figure 1. In group 4, vitamin C was given daily for 4 weeks prior to REM sleep deprivation. The physical parameters were measured and the iota was subjected for further analysis. REM sleep deprivation was induced using an inverted flowpot technique. During REM stage, the animal lose their muscle tone and fall into the water. Thus, rats wake up. From the results, in REM sleep deprivation group, the body weight gain significantly decreased despite increase in food consumption, suggested an increase in energy expenditure. An imbalance of oxidative stress markers was noted in REM sleep deprivation. Increased systolic blood pressure together with impaired endothelial dependent relaxation in REM sleep deprivation suggested of endothelial dysfunction. Histology study demonstrated structural damage only in REM sleep deprivation. Interestingly, vitamin C improved all the parameters investigated. To conclude, it is proposed that oxidative stress-induced endothelial dysfunction in REM sleep deprivation and vitamin C has beneficial effects. That's the end of my presentation. So, sleep well for better health.
Thank you very much, Dr. Liza. I welcome Dr. Shafiq to ask any questions.
Yeah, thank you, Dr. Izuddin, and thank you, Dr. Liza, for that poster presentation. I think it's quite clear based on the poster as well. I just have one simple question, or basically just something that caught my attention in your work is that were you able to identify any other biomarkers from REM sleep deprivation in induced endothelial dysfunction other than the reactive oxidative stress?
All right. Okay. Thank you for the question. Actually, the hallmark for the endothelial dysfunction is the impaired endothelial dependent vasorelaxation. So in that case, it is mediated by the nitric oxide. So other than oxidative stress, we also measured the protein expression of eNOS or endothelial nitric oxide synthase, which I didn't include in this poster. But it is what we have shown is that the expression of eNOS was significantly lower in REM sleep deprivation when compared to other groups.
Okay, yeah. Thanks. I think that's just all my question. I just need to get clarification on that part. Thank you.
All right, thank you.
All right, thank you, Dr. Shafiq and Dr. Liza. We will move on to our next presenter, the third one, Ms. Raghdaa Hamdan Zarzour from Universiti Sains Malaysia. Take it away, Ms. Raghdaa.
Zarzour. I am here today to present my poster aiming to illustrate the relationship between non-alcoholic fatty liver disease, which is the hepatic manifestation of metabolic syndrome, and Alzheimer's disease, the most common type of dementia. Since the precise relationship between the two diseases is still not fully understood, our main objective is to explore the molecular relationship between them. Our method depends on using the emerging computational bioinformatic tools. On to the results, 130 genes were associated with Alzheimer's disease and 359 were NURULUMI-related genes. Our first analysis showed that the two diseases are sharing 11 pathways interfering with autophagy, neurotransmitter receptors, postsynaptic signal transmission, and immune system. While our second analysis has shown the matching of 28 biological processes between NAFLD and Alzheimer's disease, as we can see in the figure number 2. This includes the neuronal oxidative stress, neuron death, apoptic process, and cellular response to lipopolysaccharide and insulin stimulus. Both diseases are linked indirectly by sharing the general cellular process, such as inflammation, hemostasis, and the activation of NMDA receptors involved in the synaptic plasticity, affecting learning and memory. Moreover, lipid metabolism seems to be a main key player in this cycle. This can indicate a strong direct relationship between metabolic disorders and neurological diseases that strongly warrants further experimental investigations. This is all from my side. Stay safe and take care.
Right. Admin, can you show the slide again? Because I need to see the poster.
I am Raghdaa Zarzour.
Okay. Yes. All right. So you're trying to find the link or the relationship between the two diseases, right? But you didn't really explain what is the hypothesized theory or how it is linked through mechanism or pathogenesis or perhaps a similar mechanism or whatnot. Can you explain how these two diseases are linked together?
Yes, very good morning for all the panels and for all the attendees. First of all, this one is to highlight actually the relationship and to explain the molecular, the common molecular pathways between the two diseases. Let us say that the relationship between the obesity, the NAFLD, and all the metabolic disorders is very well established for the cardiovascular diseases. But still for the mental diseases such as Alzheimer and Parkinson disease, it is still not very well known. So that's why we want to identify what are the genes which are responsible for this disease and for, let us say, for NAFLD disease and for Alzheimer disease. And then After that, we want to see the common genes between them and what are the biological processes that are shared between the two diseases. After that, we can have that target from those genes for the further, let us say, in vitro and also in vivo experiments. Because if from the beginning we want to screen all the genes, all the biological processes, it will be time consuming and also it will be costing. When we have this, I mean shared 11 pathways and also the 28 biological processes, we can limit and we see the, let us say, an MDA process. We can target this one to come up with a model which of Parkinson's disease that is resulting from metabolic disorders. Let us say we can use the high-fat diet to induce the model in vivo, and also we can see what is the effect from the point of view of pathophysiology and also from the therapeutic effects. So we can see what is the effect of the agents, for the pharmacological agents or for the herbal extracts on this, I mean, gene, especially these genes, and also to see what is the improvement that is happening due to that treatment.
So from your study, I can see that is it the oxidation reduction process, which is common between the two disease?
Actually, we have realized here that as we know, the lipid metabolism is the main problem in the the inflammatory process. Here we have realized that there is
All right. Can we have the fourth presenter, Ms. Siti Balqis Adnan from International Islamic University Malaysia, to present her research. Please welcome Siti Balqis. Admin, I can't hear the voice.
And then to assess the antimicrobial susceptibilities of the streptococcus pneumoniae and their links with specific serotypes to analyze molecular.
Alina, can restart this one?
Contributing to the antimicrobial susceptibility. Assalamu alaikum and greetings. I am Siti Balqis Adnan from IUM. I would like to present one systematic review of pneumococcal infection, diagnosis and treatment in Malaysia. We have set 4 objectives: to analyze prevalent serotypes of Streptococcus pneumoniae, and then to assess the antimicrobial susceptibilities of the Streptococcus pneumoniae and their links with specific serotypes, to analyze molecular and genotypic characterizations contributing to the antimicrobial susceptibilities, and then to evaluate the potential coverage of pneumococcal vaccine and its clinical and academic impacts in Malaysia. So the selected articles were the studies published from 2000 to 2021. These are our findings. So based on figure 1, the most common serotypes were 19F, 23F, 19A6, A6B, and 23A. Based on figure 3, we can see that there is consistency in the trend of resistance of the isolates to penicillin and erythromycin. And then based on figure 4, there is a significant association between serotype and penicillin susceptibility. The next 2 tables suggest that the varying antimicrobial susceptibilities between susceptible and non-susceptible isolates might have been contributed by alteration of resistance determinant genes. Most studies reported an estimated pneumococcal pneumococcal vaccine coverage of more than 50%. And then it was estimated that PCV13 was the most cost-effective than other pneumococcal vaccines. In conclusion, our systematic review has successfully achieved our predefined objectives of the study. We hope that this study would be of interest to the researchers and policymakers to develop an official database for the diagnosis of pneumococcal disease, and then to enact a strict regulation for antibiotic use, to conduct more epidemiological studies, and also to implement a global collaboration between countries for a continuous surveillance. Thank you.
Thank you, Siti Balqis. Dr. Shafiq, if you have any questions.
Yeah, sure. Thank you, Balqis, for that poster, and I'm sure systematic. Is Balqis here in the panel?
Yes, yes.
Oh, okay, all right. Sorry, I didn't see you. I just I'm sure that the systematic review is not an easy thing to do, but I just would like you to summarize in maybe a few sentences as well. What would be the major significance of your systematic review, and what do you think would be your next step? Is this something that you're going to do in the lab?
Okay. Can you see me?
Yep.
Okay. Thank you so much for the question. I really appreciate it. So to summarize it, our systematic review, our main aim is for a curated documentation where from our knowledge, there is no current collected documentation that has our objectives. So from our findings, we have found that the prevalent stereotypes in which these stereotypes are important to know which current pneumococcal vaccine in Malaysia can cover the stereotypes in Malaysia. And then we found the genetic characterization, genetic relationship with the antimicrobial susceptibility, and then also about the pneumococcal vaccination coverage, because the implementation is yet to be widely covered in Malaysia. Yes, so I am currently an undergraduate student, final year biomedical from IUM. So I will further my studies after this. So currently, I will target to carry this into the laboratory, and we will study on the genetic characterization, such as the modification of the resistance determinant genes. So I will aim here, and then I will target for the interest of other researchers to produce more studies with more larger geographical distribution, specifically in Malaysia. So I think this is important because as an awareness to the Malaysians, and also because as a preparation for Malaysian infectious disease rates. Okay, so that's all. Thank you.
Fantastic. So well done and best of luck in your lab.
Okay, thank you.
That's all from me, Dr. Izuddin.
Thank you.
Okay, thank you, Dr. Shafiq and Dr. Siti Balqis. Give us 1 minute. All right, next we have another presenter who's going to talk about a systematic review. Miss Jia Wen Chai, from Quest International University, Perak. Please help me welcome Miss Jia Wen Chai.
Hi, my name is Chai Jia Wen. Today, I'm presenting a systematic review of the AC methods for modified release metformin tablets specialized in extended release formulation. Diabetes is a major public health concern in Malaysia, which affects 2.8 million individuals. Among a variety of oral hypoglycemic, metformin is one of the most popular prescribed anti-diabetic drugs sold under the brand name of Glucopatch. According to the British Pharmacopoeia and United States Pharmacopoeia, the AC method that applies on the plain tablet is often not applicable to extended release formulation. Researchers believe that the fluctuation of result is caused by the culprit cross-camelosodium, which is the most commonly used excipient in the preparation of extended release tablets. as this super disintegrant agent creates metformin cross-camelose complex that causes low percentage of metformin recovery during analysis. A total of 75 records searched based on the combination of terms used, and only 5 met the inclusion criteria of studies that mention current assay method in analyzing metformin extended release tablet. Eventually, only 2 studies are included in the review. Study conducted by Huang et al. Suggested adding arginine in assay procedure, creating competitive drug excipient interaction to form arginine cross-camelose complex. By then, metformin will be freely bound and hence increase its percentage of recovery to achieve consistent and accurate test results. Study conducted by Umapati and colleagues suggested replacing water, which is a solvent used in sample preparation, to 99% ethanol and methanol. This substitution could improve the solubility of metformin into the organic solvent and hence separating free metformin from the formulation.
Okay, thank you, Ms. Jia Wen Chai. So basically, the systematic review is reviewing the assay, which looked at the metformin release. Is it for the delivery system, right?
Yeah, it's the S.
Yeah. So why do you do this? I mean, why do you need to? Why are you studying the assay? Because of course, drugs which have been marketed or used clinically, I mean, the mechanism or the release is pretty well known. So what's the purpose of doing this systematic review?
Because they found that the official method actually doesn't apply on the extended release formulation as their method for metformin extended release tablet. Of course, metformin is already marketed, but however, I think this study not only benefits for the researchers that wish to work out for the assay procedure, whereas also benefit to other drug developed researchers who wish to develop extended release formulation in future. Of course, if they have similar property with metformin or actually share the same problem in the assay procedure.
Right. So you are saying that the extended release type is not well studied?
Yeah.
Okay. Similar to Dr. Shafiq's question to the previous presenter. So what is your next plan on in pursuing this particular topic? Are you planning to do some lab investigation on this or?
Yeah, at first this is the lab assessment plan, but however, due to the pandemic, so it is restricted by our university to go for the lab procedures. So after for this systematic review, I'll go for the lab procedure to based on my results. So I'll go and
what is it that you're studying? What is it that you are investigating?
Investigate because in the result is due to the interaction between metformin and also the excipients. So there is way to so the study also applied a theory to restrict and remove the boundary of drug excipient interaction. So by practically laboratory results, so we can know is this workable or not.
All right. Thank you, Jia Wen, and good luck in your lab journey afterwards.
Sure. Thank you.
All right. Okay. So give us a minute before we proceed with the next. Okay, let's move on with our next presenter, Alexandra Leclezio. I hope I pronounced your name correctly, from Zarzour Siwu Segur Ramgoolam Medical College, Mauritius. Take it away.
Good day. My name is Alexandra Leclezio. I'm a student at SSR Medical College, Mauritius, and the research I'll be presenting today is titled Medical Students' Perspectives on Absenteeism, a cross-sectional study from a private medical college in Mauritius. The battle of attendance and absenteeism has long plagued both students and faculty alike. The aim of this study was to identify and understand the perceptions of medical students on reasons as to why absenteeism occurs, and the factors to improve absenteeism. A cross-sectional observational study was conducted from August 2018 to September 2018 at SSR Medical College. 503 students participated in the study. Factors for absenteeism were weather, lack of motivation, the need for self-studying, health problems, and lecture timetables. And South Africans noted that homesickness was a big factor that led to absenteeism. Factors that reduced absenteeism, however, were student counselling, active lecture engagement, clear and logical lectures, reduced teaching hours, and peer monitoring by classmates and senior students, especially the female cohort, were bound to improve their attendance through peer mentoring by classmates and senior students. The conclusion is that the factors that reduce absenteeism should be employed more readily, and the factors that precipitate absenteeism should be rectified and seen. This being implemented with the sole purpose of producing capable and viable graduates. Thank you.
Thank you, Alexandra. Dr. Shafiq, over to you.
Hi, Alexandra. Thank you. Interesting study indeed on the absenteeism. But I do have a few things in mind when I was looking at your posters. Is this something that, based on your findings, is this something that you can create in terms of, methodology, in terms of increased the outcome of so-called attendance or reduced absenteeism, or something that you can influence in terms of policymakers at the end in order to implement the same methods, for example, in all schools or universities? What are your thoughts on that?
Good day. Thank you very much for your question. Can you hear me?
Yep.
Okay, thank you. So yes, definitely this study is going to be used as a model, hopefully in my university, to increase attendance. And how we can increase attendance is perhaps by looking at how lecture engagement, active lecture engagement, can be used to implore students to attend lectures better. For example, which ways do students respond better to visual lectures, to didactic lectures? We can also take topics such as homesickness, which were discussed in my study, and mental health and lack of motivation, and implement that in a policy where we have more student mentoring and more supervision by teachers on the different students' mental health. Obviously, things such as weather and health problems are more difficult to correct. However, the infrastructure of the university can be better suited to the colder weather conditions that we have here in the center of the island of Mauritius. And student seminars were as a sort of incentive for students to attend the lectures, were very helpful, and we can implement that in the policy to have more student seminars.
Yeah, thank you. Just one more. Do you think by the addition of technology, for example, in this, everything is online nowadays, even this conference in particular, do you think that would give an advantage to your work as well?
I believe that we can still have more attendance if the lectures are structured in a way that the students are more receptive, even online. And we can still have peer mentoring online. After lectures, we can have one-on-one sessions with the teachers to get feedback as to how much the students understand, how much they want to attend. And this is very important because students and doctors are shaped in the classroom. And if we can solve these issues, we can produce more capable and viable graduates from the medical colleges.
Fantastic. Well done. Thank you. That's all for me.
Thank you.
Thank you, Alexandra and Dr. Shafiq. Give us a minute. All right, we're halfway there. I will now present to you Miss Chuah Candy from Utah University Tunku Abdul Rahman to present her research. Please welcome Chuah Candy.
Giardiasis is a gastrointestinal infection caused by the protozoan parasite Giardia intestinalis. Severe infection leads to chronic diarrhea and complications such as lactose intolerance. Several drugs are available for giardiasis. However, drug resistance is one of the issues that have always been reported. The finding of a novel potential drug target is important to control the disease. In this study, we examined the role of alpha-gyrdins in response to drug treatment in Giardia intestinalis. Giardia intestinalis were treated with increasing concentration of mebendazole and metronidazole. Our findings show that Giardia intestinalis that were treated with high concentration of mebendazole or metronidazole show significant reduction in viability, and they were more susceptible to the action of mebendazole when compared with metronidazole. The release of reactive oxygen species was not detected in either mebendazole or metronidazole treated Giardia intestinalis. We also found that Giardia intestinalis treated with 100 micromolar mebendazole or metronidazole show significant upregulated transcriptional expression of alpha-2-gyrdin. In short, this observation suggests that alpha-2-gyrdin could serve as a potential promising target for future drug design and vaccine development.
All right, thank you, Chuah Candy. This is a pretty difficult study for me to digest. So what is actually, can you explain a bit to us what is actually alpha-2-gyrdin gene or target that you are looking at?
Hi, can you hear me?
Yes, yes.
All right, thank you for the questions. Okay, so basically alpha-2-gyrdins, they actually belong to the family of alpha-gyrdin, which is also known as annexin E family in parasite. So this protein or gene, they shown significant protection in terms of oxidative stress in plants and also mammals, but they have not. This gene has not been reported in parasite. So in this study, we would like to check how these gene actually respond to the stress that is induced by drug response in parasite. And from our study, we managed to show that alpha-2-gyrdin seem to play a protection role in these parasites. Yeah.
Right. So your study showed it has the protective mechanism. How exactly the protection mechanism takes place?
Okay, so further study actually need to be carried out, because in this study we only examine the transcriptional level, which is the gene level, and we found that they actually show significant transcriptional level in response to drug treatment. So we need to carry out further study to actually further characterize the role of this gene further.
Any hypothesized mechanism?
So we think that this gene alpha-2-gyrdin actually play a role in the regulation of drug-induced stress in this parasite.
All right. Thank you, Candy, and well done.
All right, thank you.
Welcome. Okay. Give us 1 minute to. All right, we'll proceed with our next presenter, Muhammad Wahizul Haswan Abdul Aziz from Universiti Kebangsaan Malaysia to present his research. Please welcome Wahizul.
Assalamualaikum and good day. My name is Muhammad Wahizul Haswan bin Abdul Aziz and I will be presenting my poster entitled GCMS Analysis of Terpenoids from Extracts of Canarium Odontophilum mic dabai leaf. Canarium odontophilum mic, or locally known as dabai, is an endemic plant in Sarawak, Malaysia, and devoured as a snack food by the natives. To date, only several pharmacological studies have been conducted to investigate the properties of dabai leaf. Terpenoids are defined as secondary metabolites and they demonstrate various biological activities. Therefore, we identified the content of terpenoids to predict the biological activities of dabai leaf. The leaves were ground and extracted with hexane and ethanol, and the crudes were analyzed using GCMS. The crudes have unique physical features. The ethanol extract was dark green in color, meanwhile the hexane extract was yellowish brown in color. Both have an aromatic, strong scent. Extraction using N-hexane yielded 0.624%, while ethanol yielded 9.9%. The GCMS analysis showed the total terpenoids content were 36.67% and 14% for hexane and ethanol extracts respectively. N-hexadecanoic acid, phytol, and octadecanoic acid were the predominant in hexane extract, while only phytol and N-hexadecanoic acid were major in ethanol extract. We were able to predict the biological activities of dabai leaves extracts based on the various terpenoids detected. Further research, including the isolation and biological activity of individual terpenoids, will certainly yield rewarding findings, thus will open up new areas of research into particular compounds and their pharmacological potential in the future. In conclusion, our study confirmed that hexane extract contains richer amount and greater number of terpenoids compared to the ethanol extract. Thank you.
All right, thank you, Dr. Shafiq.
Yep. Thank you, Muhammad Wahizul, for that poster presentation. I do have several questions though, but I'm just going to make it quick and a short one. My question is, why GCMS?
Okay, actually, why I chose GCMS is when I think of I want to check for terpenoids. Actually, terpenoid is a volatile compound, so just the gas chromatography is the best method to check the content.
Okay. Do you have any plan to continue moving forward to non-volatile components like LCMS?
Because my main compound to check is actually terpenoid for my study, so I will be only check for the terpenoid which is volatile.
Okay, all right, understandable. And were you able to find any terpenoids, I don't know, based on your readings or based on your lab works, any terpenoids or any molecules from your GCMS that could have potential in terms of therapeutic applications, because that's what you're trying to identify anyway, right?
Yes. So based on my research, I have found that the three predominant terpenoid, which is N-hexadecanoic acid, phytol, and octadecanoic acid. So all three of them have biological activities claimed from the previous study, such as high in antioxidant, anti-tumor, and whatnot.
Okay, great, fantastic, well done. That's all from me.
Okay, thank you.
Right, thank you, Wahizul and Dr. Shafiq. I think he got the correct judge. All right.
Thank you so much.
Right. So we move on to our next poster presenter, NURULUMI AHMAD from Universiti Sultan Zainal Abidin, Unisa, Terengganu. Please help me welcome NURULUMI.
Assalamualaikum and a very good day. My name is NURULUMI binti Ahmad. I'm a lecturer from Faculty of Pharmacy, UNISA. So today I'm going to present my poster presentation entitled "The Effect of Curcumin on Wound Healing of Rats." So our objective in this research is to determine the effectiveness of curcumin on wound length of the wound healing of a rat, and to investigate the percentage of wound healing. So we used 21 sprague downy rats, and we divided into 3 groups: normal group, wound control group, where we did the wound incision without giving any treatment, and group C, the incision with application of 2% of curcumin paste. So we then monitor the rats on day 2, day 4, day 6, day 8, day 10, 12, and 14 after the curcumin application. So as a result, as you can see here, this will be the difference between 2 groups. The group B, the incision without curcumin paste, and group C, the curcumin with application of curcumin paste. As you can see also, there are actually differences between these 2 groups, where the wound, the percentage of wound healing is higher in group C, but when we calculated the significance of both groups, we see no differences. So the conclusion, although there are differences, but it's not so significant, and this may be due to a few factors, and further study should do a further study on this. Okay, so thank you so much. Bye bye.
Okay, thank you, NURULUMI. So, when studying the effects of wound healing, I know it's not insufficient just to measure the how to say the length of or the closure of the wound. So, what's your next step to validate your findings? Is NURULUMI in? NURULUMI? I don't think she's in. We will get back to her later. I think we'll proceed with our next presenter, which is Muhammad AFZAL bin ALIAS from IIUM to deliver this presentation. Please welcome.
Non-alcoholic steatohepatitis is a chronic liver disease where it is believed that excessive oxidative stress and inflammatory response contribute significantly to its pathogenesis. Tulang honey, on the other hand, is known for its anti-inflammatory and antioxidative effects and has been proposed as a potential supplement in the management of NASH. The study aimed to identify potential key genes and biological processes associated with the therapeutic effects of tulang honey in NASH animal model. 20 sprague downy rats were used in this experiment. 5 rats were given standard commercial pellets throughout the experimental period, which is 20 weeks, and acted as controls, while the other 15 rats were subjected to a 12% high cholesterol diet for 16 weeks to induce NASH. They were then divided into 3 groups, with each group receiving 3 different dosages of tulang honey supplementation for 4 weeks. RNA was then extracted from serum, and microarray analysis was performed. From this analysis, a total of 3,062 differentially expressed genes were identified. 2,788 were upregulated and 274 were downregulated. As we can see from the volcano plot here, the red indicating the upregulated genes and the blue indicating the downregulated genes. From these genes, 15 were significantly upregulated, including aldehyde dehydrogenase family, cytochrome P450, and nuclear transcription factor, whilst 13 genes were significantly downregulated, such as apelin, interferon regulatory factor 2, and sterol coenzyme A. Following pathway enrichment analysis, we found 34 biological processes that were significantly modified. Among the pathways implicated in the effects of tulang honey are those involved in inflammatory response, lipid biosynthetic processes, and cholesterol metabolic process. The present study demonstrated that the differentially expressed genes involved in the therapeutic effects of tulang honey were enriched in several pathways, and further validation is required to confirm the specific roles. Thank you.
Thank you, Afzal. Dr. Shafiq, over to you.
Yep, thank you. Thank you, Afzal, for that poster presentation. Just a few confirmation from you on your part. Do you have any plan to further your study, to further this particular study into looking something more specific, like a specific pathways? For example, we're talking about proteins, proteomics, because one gene can simply give rise to many proteins as well, and proteins can be an important factor in any pathways, I think, as we all know. So what are your next plan after this, basically?
Yep. So can you hear me?
Yep.
Yep. So basically, we're looking specifically for NASH. We want to look into the inflammatory pathways and also the fibrosis pathway, since NASH involves fibrosis of the liver. So we want to look at those two pathways and further on, and go further on from there.
Okay, great. And also the tulang honey is just it's a crude application to the animal model as well, is it?
Yes, it is. Yep.
Okay, and were you able to identify any specific components from the tulang honey itself from crude?
Yeah, so we that's the next step. We haven't done that specifically. We bought commercial tulang honey and we use that in experiment. But further on in the experiment, we'll look into the specific contents of tulang honey.
Yeah, that'd be interesting as well, because then we can identify specific components in the honey that can contribute towards all the upregulation and downregulation of the gene as well. That'd be great.
Yep, definitely.
Okay, all right, thank you. That's all for me.
Okay. Thanks.
Thank you, Afzal. We are down to our last two presenters. The first one, Ibrahim Abuga from International Islamic University, Malaysia. Mr. Ibrahim, please take it away.
Assalamualaikum warahmatullahi wabarakatuh. My name is Ibrahim Abuga. I'm here to present my poster presentation. The title of my research is In Vitro Antibacterial Screening and Phytochemical Profiling of Malaysian Traditional Plants Used for Treating Diarrhea Infection. Introduction: antibiotic resistance associated with diarrhea disease is a serious case that remains a stimulating situation to health professionals in developing and developed nations. Methodology: the methodology adopted for the antibacterial screening was microdilution growth acid using 96 well microplates. For the total phenolic content, the following calcium acid was employed, and the extract were evaluated for their chemical constituent using liquid chromatography with tandem mass spectrometry. Result and discussion: the least minimum inhibitory concentration value of 7.812 microgram per ml was observed with the leaf extract of urea quality against Staphylococcus aureus. The result is in agreement with the findings of Lua and Prasad 2010, reporting good antibacterial activity of the methanolic leaf extract of the plant against the clinical isolate of Staphylococcus aureus with MIC value of 12.5 microgram per ml. The extract of Zingiber officinale possesses the highest TPC of 445.22 microgram garlic acid equivalent per milligram. This is in conformity with the findings of Maidul et al. 2011 that showed a total of 101.6 plus or minus 0.6 mg garlic acid equivalent per 100 gram juice extract of the plant. The compound Vescoaminoin C was found to possess antibacterial activity against some species of bacteria. In conclusion, the result obtained from this study disclosed vital scientific evidence of the traditional uses of plants in treating diarrhea infection.
All right, thank you, Ibrahim. Are you there, Ibrahim? Right, so this study is quite general in the sense that you're basically taking compounds from various plants and looking at the phytochemical and antibacterial studies. So these compounds that you chose of plants, how do you, I mean, why are they chosen, and how do you choose these compounds? And is there any control, a positive control, which you can compare that the efficacy with?
Assalamu alaikum. Can you hear me?
Yes, I can. But your voice is not that clear in the presentation just now.
Okay.
But I can hear you now.
Okay. Actually, the plants were selected based on their traditional records. Based on their traditional records, which express various parts of the plants that we are used in Malaysia to treat various infections. So in a nutshell, the plants were selected based on their traditional usage in Malaysia to treat different infections, especially infections that are related to diarrhea. Exactly.
Right, and the control that you use in comparison, do you have any control?
Yes, I use tetracycline.
Okay.
Yes, tetracycline was used as a positive control.
So what next? Because basically this is just like a screening. So what next for you?
Yes, exactly. My next attempt is to look into the molecular aspect of the this thing, of the antibacterial effect of the most potent plants on the molecular aspects. For example, the gene expression, maybe some genes that are vital for the antibacterial effect will be selected and then will be tested against the most potent plant extracts.
All right. Thank you, Ibrahim. Excellent. And good luck. Thank you, Ibrahim. Give us a minute for us to score the presentation. Okay, we are down to our final poster presenter for the day, Mr. Maidul Islam from Apex Biotechnology Laboratory, Bangladesh. Please help me welcome Mr. Maidul.
I'm Maidul Islam from Bangladesh. I would like to talk about a single-center study outcome briefly, which was for evaluating the potential and efficacy of some modern research medicinal foods as well as medicinal herbs for diverse cancer management. Globally, cancer is the leading cause of morbidity and mortality. In background study, we found that due to conventional therapy resistance, high toxicity, and lack of targeted specificity, globally researchers and physicians are gaining frequent attention to chemosensitizing bioactive compound for their multi-target potential and relatively low toxic manner. Our study center was Dhaka division-based center comprised of naturopathic doctors and researchers. Patients from different corners of Dhaka division were assessed, where patients were routinely followed up by face-to-face contact, sometimes over phone asking manner. Study conducted only to measure their signs and symptoms, easing as well as improvement of their quality of life. No post-management, pathological and medical diagnoses were done. Results found as reduction of tumor volume, malignancies, vaginal research, and improving of their quality of life. In this study, we try to secure for preliminary understanding about the potential and efficiency of different natural compounds to match the belief and reality of naturopathic treatment level. Conclusively, we can say that modern research medicinal food showed what is positive outcome from diverse cancer management. Further, post-management pathological and molecular marker identification and measurement is needed for safe and effective anti-cancer drug development. That's all. Thank you.
Thank you, Mr. Maidul. Dr. Shafiq, your final questions?
Okay, hi, Maidul. Thank you for the presentation. So I hope Maidul is in here. Okay, all right. So I just have a few things running in my mind. So basically, the idea of your work is to create a supplement. Basically, that's a supplement, supplementary, not a proper medication for this particular cancer patients as well. Is that what you're trying to aim from your work?
Yes, sir. Can you hear me?
Yep.
Hello.
Yep, I can hear you.
Yes, sir. Just our task was to assess the different bioactive compound from different superfood, like as just Moringa citrifolium, just spirulina, just Ganoderma lucidium, and so there they have a huge potential to treat cancer as well as management of different cancer. Just we tried here in a single center study to find out the efficacy of their bioactive compound, especially for sign symptoms management and improve their quality of life. Here we finally found that several patients of different cancers, for example, breast cancer, fibroblast, just ovarian cancer, just leukemia cancer, and sometimes colorectal cancer patients are getting better, getting better their quality of life, concerning their quality of life, and we found it, and we hopefully will be able to just evaluate their different just different potential effect on diverse cancer management.
Okay. How do you prepare the superfood mix? Is it orally administered or is it a part of the patient's diet?
Yes, just we just ask those patients to take their supplement, just food supplement as a food supplement. Yes.
Okay. All right. Great. So just one last short question. Were you able to identify any bioactive compound from the herbs that you are using in your work that may contribute towards the improved health condition of the patients?
Just as you know that just we didn't do any.
Yeah. Have you done any? Yes, any molecular study on the plants, on herbs itself? Were you able to identify any bioactive components?
Yeah, hopefully next time just we will be able to identify several miraculous bioactive compound from those supplements, especially for just getting emphasized on four bioactive compound here, just from Loli, just Moringa citrifolia, just spirulina and Ganoderma lucidium, and another just miraculous herbs from Malaysia. Sibyl is the Christia vespertilianis. It's a miraculous herbs, and it's having different types of bioactive compound for different cancer management.
Okay, fantastic.
And we'll try further to identify just a bioactive compound. Yeah, that's all.
Sure, all good from me. That's all from me. Thank you, Maidul.
Thank you, both of you, Maidul and Dr. Shafiq. So that's our last presenter. Before we conclude, perhaps is NURULUMI, has she joined in? Has NURULUMI joined in for to receive a question? So she wasn't there earlier on. I think she's still, is she here?
No, we couldn't find it.
All right, never mind. So well done to all poster presenters for this session. I appreciate if all of you would open your camera and we could take a group photo. Dr. Farouk, you'll do the counting and taking the pictures, I believe?
Yeah.
Okay, cool.
Can I also request others, presenters? We Dr. Satira can also on, and Ibrahim. Let's start video. So AFZAL. Okay, just give me a second. All right, smile, 1, 2, 3. This is first shot. Let me take another one. Okay, smile again. 1, 2, 3. Great, thank you.
All right, congratulations once again to all presenters of the day. Don't forget to come back at 2:30. We have another parallel session for symposium. I think it's cancer research as well as cardiovascular research. Okay, so if you're interested with cardiovascular studies, you can join room 1. And if you're interested with cancer research, you can join room 2. We have 2 fantastic speakers, Associate Professor Dr. Satira from UKM and Dr. Alan Yu Wahdi from Monash. All right, I'll see you guys again at 2:30. Do come back, have a good rest and a good lunch, and I'll see you guys soon. Thank you very much and well done, everyone.